working from Josh's project. So let me first show you how we got here. So start out with, I just Googled PubMed, because that'll take us to NCBI. So you may have worked on PubMed here on NCBI. And so we're accustomed to just typing in the name of, I don't know, if we want to, whatever we want to learn about here, and it'll do a search for us. But actually, if we look here, right next to it, we see we have a whole bunch of different databases. So some of these may also be familiar. We've used nucleotide databases or protein databases to search for stuff. So now we're going to use a couple that are a little bit less familiar. So the SRA is for short read archive, and that's where a lot of next generation sequencing data is stored. So we could actually go directly there and search for our organism of interest, which in this case is, which I've forgotten the name of. I think it's Plasmodium vincii. All right, it worked. And that works pretty well. As you can see, it's pulled back some things. We'll get here in a moment. An even better way to do that is to go to the taxonomy page. So if we search for taxonomy now it's actually, it finds the page in all of NCBI that stores all the data for this one species. And so if we click through, what we see here is it gives on the right, it's a summary of all the data that we have. And so at the top, it says there are 5,400 different nucleotide entries, which could be a whole chromosome or it could be just a short little sequence, and the number of proteins that are in the database, the number of genomes, and so forth. So this is great to play around with. I highly recommend it. You'll find all kinds of cool stuff. But what we're going to do is go down to the short read experiments. So we're interested in the short read archive. So if we click on this, this is going to take us to this page where we have different experiments. So we see 2 entries here. And so let's look at each one of those. So what this means is this is a single data set that was generated. It was generated by an Illumina sequencer, specifically an Illumina HiSeq 2000. And this data set consists of 40 million, 39.6 million spots, or that is different sequences, different reads, totaling 8.3 gigabases. And so that's cool. And we see that there are 2 of them. And we'll have to, on the one hand, there's a lot of information here. On the other hand, you see there's not a lot of information. It just tells us that it's RNA sequencing. So if we click on one of them, it'll give us a bunch more information about it. And so here we have a description of it. And so this is just some ID. This is what we saw before. And then this design tells us basically what they actually did. So Illumina sequencing, that is sequencing using an Illumina sequencer of this species, Plasmodium vincii, via random selection. So random selection meaning that trying to sequence all the RNA in the genome. And then the study said it's RNA-seq portion, the Plasmodium 100 Genomes Project, so they're sequencing 100 different Plasmodium species. So that's just the project that generated this data, right? Because somewhere, some humans actually generated this data. And so we have all this other information. So this stuff is great to explore. But if we want to just get this data, we can do that using this tag right here. This is the ID of this run. That is, they ran the sequencer, and it generated this run. Remember, 39,600,000 different reads with a total size, and then basically when it came out. So we're going to just grab this, and then we're going to go back over to the cluster, and we're going to figure out how to get this data. So it turns out that there's a program that will just go grab it for us. It's called FastQDump. And we just type in FastQDump in the name of our file. And what's happening now, the reason that it's hanging, is in the background, it's actually going and grabbing the sequence. So this is quite a big file, so rather than letting the whole thing run through, I'm going to cancel it. and we can actually just look at the file that's been created. If we look in our directory, we'll see that now there is this file, which is the thing that we inputted, this sequence ID .fastq. So this file now contains the raw data that we want. So we can look at it. And what we see, so we see all this data. So let's unpack this. So actually what we're looking at here is a series of 4-line entries. So this first 4 lines, I know it looks like 6 lines, but that's because these 2 lines are just running over. It's just folded over. So this whole thing is just 1 line. So these 4 lines is basically one read. It's one nucleotide sequence that was observed. And so that nucleotide sequence that was observed is NGAA, GGCAT, and so forth. And we find that overall it's 210 nucleotides. So this is just telling us the random name of this read. This gives us the sequence of the read, and then we have the random name of it again. And then we have, these are the quality scores. So right now I'm not going to dwell on that, but that basically tells us how good of a sequence this is. Sometimes they're very low, like all these hashtags down here. These hashtags say that this is low quality sequence. So this hashtag means that this G is a low quality base. And this hashtag means that this C is a low quality base. That is that we're not very confident of this region that that's really the sequence. It might be wrong. So that's a little bit scary, but fortunately the programs that people have written to analyze these huge amounts of data, they take that stuff into account. So we don't need to worry about it particularly. But, so long story short, this 4th line is telling us the quality scores. And I think probably for that tutorial, that we'll call that a tutorial. So that's a tutorial on how to find and download data from SRA.